Hello and welcome to this TATAA Biocenter seminar on sample collection and extraction of nucleic acids. My name is Robert Sjöback and I come from TATAA Biocenter. TATAA Biocenter was founded in 2001 as a spin-off from Chalmers University. Originally giving training courses in real-time PCR, but today mainly laboratory services for other customers, both within the industry and the academy. Today we also use a lot of other techniques like next-gen sequencing and protein analysis. In addition to that, we also have our own product development and we distribute several products within the area of nucleic acid analysis. Finally, we also participate in several publicly funded research projects. Today I will talk about the analysis of nucleic acid. If we look at the total analysis process, it goes through three different phases: the pre-analytical, the analytical, and the post-analytical phase. Historically, most attention has been given to the analytical phase, and a large degree of the development, both within reagents and instrumentation, has occurred here. This means that this is also a relatively precise and a very highly regulated part of the whole process. Less attention has been given to the pre-analytical test, even though it has been several publications showing that probably the largest part of the total error in the analysis occurs here. In many of these publications, the error in the pre-analytical step accounts for more than 50% of the total error. In some cases, as high as 70 or even 80% of the total error occurs here. If we, for example, take a blood sample and we want to measure a gene expression pattern in that sample, we collect the sample in an EDTA tube. We will of course have some cell death, we will have some degradation of some RNA, but the remaining cells will be intact and they will feel that they are in a different environment than the normal in the bloodstream. So the gene expression machinery will start to work and some genes will be upregulated, others will be downregulated, and the gene expression pattern will not be the same as it was in the patient originally. This is of course very easy to misinterpret. So some 10 years ago there was an EU project who started to look more intensely into the pre-analytical step. They tried to identify which parameters was most crucial to get a reproducible and robust analysis. They looked at different analytes like RNA, DNA, proteins, and metabolites, and they did look at different matrices like blood, tissue, urine, and saliva. And this resulted in 9 SEN technical specifications and later also 8 ISO technical specifications. They looked at all the processes on the pre-analytical phase, like sample collection, transportation, sample receipt, storage of sample, extraction of sample, and quality control of sample before the analytical step. Now, we will talk a little bit more about the sampling. So what do we need to consider when we talk about sampling? Well, first of all, the amount of starting material. It must be enough for all the experiments you're planning. And how much that is, well, that of course depends on the downstream application. In some cases, you have special consideration, like you may have very small nucleic acids, you may have viruses, and so on. And the amount of material must also be in the range for the kits you are using. Could be the collection kits, the extraction kits, and even in the analytical steps. In some cases, you need to do further optimization. Sometimes it's important when you take your sample. If it's bacteria, then you may want to collect them in a certain part of the cell cycle. If you have food intake or if you have medication intake, it may be important to know when you have the optimal activity of that you want to measure. Further, you want to avoid contamination as much as possible when you collect your samples. Also, sometimes you have very heterogeneous material, typically like tumors, sometimes in forensic samples and in environmental samples. If you have that, you have to increase the number of replicates you take from the sample. Otherwise, you will not get a good representation of the material you're measuring. And you want to be able to collect your sample and treat them as similar as possible. And how do you do that? Well, you should always use the same collection method. And you should try to standardize that collection method as much as possible. You need to be able to have the same temperature and condition of your samples, and preferably store them the same time. Further, when you collect your sample, you normally need to stabilize the sample as soon as possible. One way of doing that is using flash freezing, but that typically requires the access of liquid nitrogen, and then you have to store your sample in -20 or even -80 freezers. You typically need dry ice to transport your samples and so on. In many cases, there are commercial stabilization solutions that can be used to collect and store your samples even at room temperature, and that may be a good option. In some cases, you have very special application, and then perhaps you need to make special preparations for those. Now I will change and talk a little bit about extraction. Why do we need to do extraction? Well, there are several reasons. First of all, we need to make the target available. Our nucleic acids is typically within some kind of cell and even in some kind of other matrix like tissues or feces or soil or whatever we are analyzing. We also need to get rid of inhibitors and other interfering substances that can affect the downstream applications. Sometimes we need to remove fluorescence contaminants. If you, for example, analyze a cell culture by qPCR, in qPCR we are measuring the fluorescence. If it's gram-negative bacteria, they typically lyse enough by heating, and the first step of the qPCR is heating to 95 degrees to activate the enzyme. That will be enough to release enough of nucleic acids to do the quantification. But most of the cultivation media contains fluorescent contaminants, so you cannot take the cell culture directly into the qPCR because these fluorescence contaminants will interfere with the reading of the qPCR. You also need to preserve the target integrity, most typically when you're analyzing RNA. Cells contains RNases, and when the cells burst, these are activated, and you need to get rid of these RNases to keep your RNA intact. Also, in many cases, you need to concentrate your target. If you have a very diluted target, then it needs to be concentrated to get a high enough concentration to be measured efficiently in the downstream application. But also extraction steps needs to be validated and controlled. And one way of doing that is using spikes. Spikes are non-target nucleic acid that we add to the sample before the extraction, and an equivalent amount to a water sample that does not go through the extraction, and we compare the result of the two. That way we can get an estimate on the yield of the extraction and also if we have any inhibition that goes through the extraction. Now, if we look back at the whole procedure of the analysis, we have just scratched a little bit on the surface of some of the parts of this procedure. If you have any questions for me or my colleagues, please come to the TATAA Biocenter booth and you can talk to me and my colleagues in the chat there. Thank you for your attention.